Example 157.4, orange trees at a citrus farm near Orlando were randomly assigned to one of 3 new fertilizers, or the traditional fertilizer being used already. The new fertilizers are supposed to produce heavier oranges. Researchers tested the claim that at least one of the new fertilizers produced a heavier orange. The following results were produced using Minitab, a statistical software package. Interpret the output. All right, so you can see the null hypothesis for the experiment was that all the means are equal. The alternative, of course, is that at least one mean is different from the others. Significance level here for the test is 5%. They say equal variances were assumed for the analysis. That is because that is the assumption underlying the ANOVA CRD experiment. So whenever you're doing a completely randomized design experiment and processing the data using ANOVA, the assumption is that the variances are equal for the different populations where the data comes from. So let's look further down here. It says that they have factor levels, values, fertilizers. There are 4 values, A, B, C, and O for original, right? So fertilizers A, B, C, and then the original fertilizer they used. You can then see the analysis of variance output. So you have the source of variation, fertilizers, error, and total. You have the degrees of freedom for each. If you notice, there are 4 levels of the treatment. So there are 3 degrees of freedom for that, 1 less than the number of fertilizers. You have the sum of squares, the adjusted sum of squares, the test statistic, and the p-value for the test. And then if you scroll down further, you can see that they've actually provided you with the means for each of these fertilizers, right? The standard deviations, and then you have confidence intervals accompanying each of those. Beneath that, you have confidence intervals plotted here for each of the fertilizers. And so basically what they're trying to do is to tell you where the point estimator was for the fertilizer and then the interval limits that are surrounding that point estimator. And you have that for each of the 3 fertilizers A, B, C, and also for the original fertilizer O. Let's try to look at the output then collectively and see if it all makes sense and if it all fits together. So let's start with that ANOVA output they provided us. All right, take a look at the F value that they gave us here. That's less than 1. Remember, an F test stat that's equal to 1 would imply that there is an equal amount of variation coming from the treatment and the error. And so if a number is less than 1, an F value is less than 1, that's going to indicate essentially that there's more variation attributable to random error than there is to the treatment group. So that's why the p-value here is so large. The p-value here is 42.7%, which of course would indicate that we would not reject under any usual alpha that we'd ever use, right? Remember, you only reject the null hypothesis when the p-value is very small, smaller than the significance level. Well, the significance level here is just 5%, so it's clear that we would not be rejecting the null hypothesis here because that p-value is definitely not less than 5%. So that means we cannot reject the null hypothesis, and remember what the null hypothesis says. It says that all the means are equal. So essentially, the p-value is large, so we can't reject the null hypothesis. So it's indicating that the treatment effect is not significant. And that means that all the fertilizers seem to work the same. Now, the fertilizers included in the study, again, are A, B, C, and O, O being the original one, right? So it seems like the original fertilizer works just as well as the 3 new ones. Now, you can see over here that they gave us basically a summary of the means, right, for the different fertilizers. And you might notice that the mean for the original fertilizer is actually the smallest mean in the group, correct? But it always comes down to that question, is it significantly smaller than the other means in the group, right? So to look at that, we're going to have to look at the confidence intervals. It's a little bit hard, though, to see it in this list of confidence intervals. It's probably better to pay attention to the drawing they gave us, right? So remember, overall, our ANOVA analysis concluded that there was not a significant difference. So if we actually take a look at this plot of the confidence intervals, you can actually see that same thing is conveyed, because in each case, the intervals overlap with one another. And for example, take fertilizers A and B. You can see that there's plenty of overlap between these confidence intervals for A and B. For example, all of these places, right? That whole area, this whole region from here to here, is where fertilizers A and B's confidence intervals overlap. And remember, the mean for fertilizer A can be any number in its interval, right? So it could be any number along this piece as well, but that's also true for B. It could be any number in the entire interval, including all the pieces I just circled there in red. So it's very possible for the true mean for fertilizer B to be in this region, right, of its interval. And likewise, it's very possible for the mean for fertilizer A to be in this region of its interval. And of course, if that were the case, it would be impossible to say if the fertilizers produce different mean yields. And that would be the same for B and C, right? There's overlap again on that vertical scale, right? All of this area they share in common along this vertical axis, right? And then of course the same is true for the original fertilizer, O. If you compare it to any one of the fertilizers means in this diagram, you'll be able to see there's overlap for all of these intervals, right? With O. So O goes from here to here on the vertical scale, and you can see that that overlaps with A here, overlaps with B here, and it overlaps with C here. And so because of that, it has overlap with all of those intervals, which means that if you compare O to any of those individual intervals, we're not able to conclude that there's a significant difference between O and those other fertilizers' intervals. So essentially, this drawing or this diagram here supports what the ANOVA hypothesis test was saying, which is that we cannot reject the null hypothesis, and as a result, we cannot conclude there's a significant difference between these different fertilizers. So again, just to recap, because the p-value is large, we're unable to reject the idea that the means for the 4 fertilizers are all equal to one another. So this data does not allow us to overturn that statement. We're not able to reject that hypothesis that all the means are equal. Perhaps maybe with a larger sample size we'd be able to, because you do see that this mean is smaller than the others, but it's not significantly smaller.